Hi everyone. It has been a very wonderful day with lots of knowledge, and I will try to top it off with some cake, as I'm the last speaker, I suppose. I come from Norway. My name is Bikal, and I'm a researcher at Norwegian Research Center in the group Gene Technology, Environment and Society, and we are also the competence center for biosafety and biorisk assessment in Norway. And I'm very happy to be here to present some of the knowledge, and thanks to Barbara for this wonderful opportunity. And in today's talk, I'm going to provide some brief information at a very basic level how we researchers use the information from plant and pathogens to develop strategies to control disease in the field. That's why the title of my presentation is Translating Pathogen and Plant Omics to Control Crop Disease. And let me remind you that by the year 2050, the population of the world is expected to be around 10 billion. And to meet the food demand of that population, we have to increase the production by 60% than that of today. That was stated in the International Year of Plant Health 2020. So, protecting plants is very crucial to protect life. And we have come across a lot of devastating disease caused by pathogens in the field that have led to great effect, devastating effect like Irish potato famine that we all are aware of. Plant pathogens like fungi, oomycetes, virus, bacteria causes devastating crop disease. That's why plant disease is a serious threat to global agriculture. And as today's theme is based around potato and leaf blight disease caused by Phytophthora infestans, I will try to give you an overview how Phytophthora species cause disease in the field. So as you can see, Phytophthora species have zoospore. They are motile, they move around. So the zoospores are produced in sporangia and they get released from the sporangia, as you can see in the video. And once these zoospores are released, they are attracted towards plant surface because plant surface or plant parts release phenolic compounds, and by chemotaxis, that means zoospores are attracted to those chemicals to absorb nutrients for their growth and establishment as a pathogen. So once these zoospores are released from the sporangia, they approach plant surface where they insist and develop germ tube. This germ tube forms hyphal swelling that try to slice the plant surface, and it helps the germ tube to get entry into the plant or host. And once it gets entry into the plant, then it establishes itself, or it tries to grow and forms host area that helps to invaginate into the plant cell, and it absorbs the nutrients from the plant that helps its growth and establishment into the host. And once it is established, it again produces sporangia from which the zoospores are released and the infection cycle is repeated. So now let us understand about plant-pathogen interaction. In nature, when plants are attacked by pathogen releases virulence factor or molecules called effectors to establish infection. But on the plant side, plant also have developed defense system. Plant has basically developed two layers of defense system. The first one is activated when pathogen-associated molecular patterns. Let me just show you by the pointer. The first layer of defense response in the plant is activated when pathogen-associated molecular patterns, or also called PAM, are recognized by the pattern recognition receptor from the plants. And when they are recognized, the first layer of defense response is activated. This response includes callose deposits, reactive oxygen burst, or cell death that tries to inhibit the infection, that tries to stop the infection from the pathogen. But then pathogen can release effectors. Effectors are the molecules released by or secreted by the pathogen that interfere with the cellular processes of the plant or modulate the defense system in the host plant. And once these effectors are released, then that again helps to trigger the infection, or it triggers the susceptibility in the plant, and that is called effector trigger susceptibility, and that allows the pathogen to grow in the plant or increase the infection. But some of these effectors can be recognized by the resistant protein in the host plant. That is the innate immunity in the plant. Plants have some resistant protein, and once this resistant protein recognizes the effectors from the pathogen, then that triggers again an immune response. That is the second layer of defense response from the plant side. And once these effectors are recognized by the resistant protein, there are some defense response like programmed cell death or hypersensitive response that tries to stop the infection, so it doesn't allow the pathogen to grow over or spread over the plant. But these pathogens are very smart. In course of evolution, they undergo selection pressure. And in a way, these effectors that were recognized by the resistant protein from the plant, they undergo changes in their sequences to avoid the recognition. And once they are not recognized, then again the infection can happen. So that again triggers effector trigger susceptibility. So this game goes on and on over time. So how to control plant disease? So to control the plant disease, we have to understand plant-pathogen interaction that I just described about. We have to understand the plant disease and the defense mechanism from the plant side against the pathogen. So how do we do that? Here comes the omics studies. Omics studies offers the required tools to understand plant response to against the pathogen stress or against different biotic stress in the plant. So what is omics? So omics refers to the sum of molecules or constituents in the cell. And the science of omics involves identifying and quantifying those molecules to understand their structure, their function, and interaction at a biological context. The science of omics involves different disciplines, like for example, transcriptomics, metagenomics, genomics, proteomics, and etc. We heard about metabolomics, and we also heard about spectronomics before. So the science of omics help us to gather those information from the plant and pathogen and use those information to understand the plant-pathogen interaction at different ecosystem. And we can use those information to develop strategies to control disease. And today in my talk, I will give you some examples how we use those information to develop strategies to control plant disease in the field. So 15 minutes talk can be very short to describe all the omics technologies. So I will be focusing on two of the omics technologies that are widely used, transcriptomics and genomics. By transcriptomics, it means that we use RNA sequencing, we study the sequences at RNA level, and that helps to understand the regulation and expression profiling of genes at certain biological state. And genomics means the studying of genome sequences, and that helps us to understand the structure and functional characteristics of genes and genetic variants. And by using transcriptomics and genomics, we can identify pathogen virulence factors, we can identify resistant gene, we can identify susceptible gene in the plant, we can also identify resistant plant varieties. And we can use this information to develop strategies, for example, integrating the resistant gene into the susceptible plant, using the resistant gene for resistant breeding, also silencing the virulence gene of the pathogen or knocking out the susceptible gene from the plant side. And this helps to control plant disease in the field. So for an example, identification of virulence factor of pathogen by using transcriptomics. This is an example in tomato. So in this example, there was comparative transcriptomic study in susceptible and resistant tomato varieties, and a gene was found that was differentially expressed while infecting the susceptible tomato varieties. Botrytis cinerea CGF1. This gene was differentially expressed. That means it was highly expressed to cause the infection. And it was considered a potential virulence factor from the pathogen side. And to validate that, this gene was silenced or mutated in the pathogen. And once this gene was mutated, the virulence of the pathogen was reduced. So that helped to validate this was a virulence factor of the pathogen. And once we know that, this information is very important to develop strategies to control the pathogen. So once we know this factor is a virulence factor for the pathogen, and this is really important for the pathogen to cause the disease, we can use this information to control the disease by using some mechanism, for example, RNA interference. And RNA interference is a mechanism just like vaccination during COVID pandemic. I think all of you got the vaccine that helped to boost your immunity. So we can use similar mechanism to control the disease by using RNA interference. And that can be used by two methods, host-induced gene silencing or spray-induced gene silencing. Host-induced gene silencing means that the virulence factor that we identify, we can integrate that into the plant through transgenic approach. Or spray-induced gene silencing is the approach where we just spray that virulence factor into the plant just like spraying pesticide. And this happens by using RNA, messenger RNA. So the mechanism is that this mechanism involves the sequence-specific recognition of the messenger RNA that directs through ribonuclease cleaving approach to degrade the messenger RNA from the pathogen virulence factors. So this happens by, there is an enzyme in cytoplasm that is diceroid enzyme, so that clips the double-stranded messenger RNA, and then it changes the double-stranded messenger RNA into a small interfering RNA that gets attached to RNA-induced silencing complex in the cytoplasm, and then one of the strand gets left behind and one strand remains in the RNA-induced silencing complex, and Argonaute protein in the cytoplasm directs it to the to the strand of messenger RNA from the pathogen virulence factor, and then it helps. Then there the magic happens. It helps to degrade that complementary strand from the double-stranded from the messenger RNA of the pathogen. Just like vaccination, how the mechanism of vaccination works, it's just like that. So it allows the degradation of the virulence factor from the pathogen so that the disease cannot happen, right? So these two mechanisms are used, and there is an example, silencing of Phytophthora infestans virulence gene PIZBP1, that is very important for Phytophthora cell maintenance by introducing the same virulence factor in potato. So it recognizes and degrades that virulence factor so the disease cannot happen. That's how we use the information from the pathogen to control the disease in the field. So another example is that using transcriptomic study to identify the genes that are involved in regulating plant immunity. So by using transcriptomics approach, we can also identify potential resistant gene in the plant. In this example, there was a comparative transcriptomic study between susceptible potato plant and resistant potato plant. And somehow, some genes in the resistant potato plants were highly expressed to combat pathogen inoculation or pathogen infection. So by doing the analysis, some potential genes were found that were only expressed in the resistant potato plant and not in the susceptible potato plant. So that gives an indication these genes might be the resistant gene. So to validate that, those genes were overexpressed in the susceptible potato plant, and after overexpressing them, the pathogen was inoculated there, and there was a combat between the potential resistant gene and pathogen inoculation, and there was hypersensitive response or cell death that helped to inhibit the infection. That's how it is confirmed, okay, this gene might be the resistant gene. So once we identify this information, or once we know these genes might be the potential resistant gene, we can use that to integrate into the susceptible plant to control the disease. So another approach to find resistant gene is also to use the effector omics approach. By effectors, I previously mentioned, effectors are the molecules secreted by the pathogen that can interfere with the cellular processes in the host plants or interfere with the defense mechanism of the host plant. And we can use the information from effectors to identify potential resistant gene. How we do that? We use this concept. I previously explained that some of these effectors can be recognized by the resistant protein in the plant. So there is second layer of defense response activated, and that causes hypersensitive response or cell death as a response. So to use that information from the effectors, we clone the effectors from the pathogen into the plant expression vector, we transform them into bacteria, and then we choose the right colonies, we make some bacterial suspension, and we inject that suspension through agro-infiltration in the model plant like tobacco. So in a study by Vivian in Wageningen University, fortunately I was there, I worked with Vivian in that group in some similar research project. They did a study where they identified some effectors of Phytophthora infestans, and then they expressed those effectors transiently in the potato plant itself. So they used this PBX infection method and found that three effectors, here are the three effectors, they induce cell death. They induce cell death in contrast to the negative control, and this is a positive control they use. So three effectors induce cell death, and it felt like, okay, these effectors might have an effect to induce the defense response through resistant protein in the host plant. And this was resistant plant, susceptible plant. In susceptible plant, the effectors has no function, but in the resistant plant, these effectors are being recognized by potential resistant protein. So they did a comparative genomic study between the resistant plant and susceptible plant and found that one of the gene was very dominant in resistant plant that led to the discovery of one of the resistant gene RPISTO1 from the potato cultivar Solanum stoloniferum. So after finding that potential resistant protein or potential resistant gene, they integrated that gene in susceptible plants. So this all 4 plants are susceptible plant, but they integrated the resistance gene in that plant. And after integrating the resistance gene in the susceptible plant through transgenic approach, they inoculated the plant with Phytophthora infestans, and there was no infection. So that helped to validate that, okay, this gene is a resistant gene and it can help the plant to be resistant against Phytophthora infestans. In contrast, the untransformed plant is completely diseased. And this is a control plant which is already resistant against Phytophthora infestans. So that's how we validate, okay, some of the genes are resistant and they can have resistant effect to combat against the pathogen. So once we identify resistant gene, or once we identify a susceptible gene in the plant that is responsible for making the plant susceptible to disease, we can use this information to change the phenotype of the plant or to change the fate of the plant to combat against the pathogen. So one of the very famous technique these days is gene editing, and we use CRISPR-Cas technology for editing the fate of the plant to make it either resistant or drought tolerant or having the features that we want in the plant. So CRISPR-Cas is a very precise gene editing technique, and once we know that, once we identify certain genes that are, for example, susceptible against the pathogen, we can knock them out so that the plant become resistant to the disease. For example, in a study, Blight et al. Found that SLMLO1 was responsible for powdery mildew in tomato. So they knocked this gene out by using CRISPR-Cas method, and once this gene was knocked out, then the tomato became resistant to powdery mildew, and not only to powdery mildew, it also became resistant to yellow leaf curl virus. So that's how we can use the information about information from the plant side to make it resistant against the disease. And the same mechanism we can also integrate resistance gene into the susceptible plant and make it resistant. But this technology also comes with ethical hurdles, because these days there are lots of controversies about GMO, but I hope most of you know that a few weeks back or a month back, EU had accepted to negotiate to change the definition about GMO, or maybe they decided that the plant that are developed by changing the gene 1 year to 20 base pairs can no longer be GMO and they can be considered just like they can be in the market without even labeling. But that's not accepted by everyone, and there's a lot of controversy about that, and that's where a current project that I'm currently working on called Darwin knocks in. The importance of Darwin project is that we are developing methods that helps to detect the plant products derived from using new genomics techniques to provide a choice to the consumer or to provide a fair food system. Because we want consumers to know what they are consuming, or we want to provide consumers a choice whether they want to consume certain product or not by providing the methods and labeling to each product, which is not currently yet in the EU at the current scenario. But yeah, it takes time to develop the methods, and we are currently developing methods using rice and tomato mutant lines. So far, we have already become successful to develop methods that can detect mutant rice. I did that just before coming to UCC last month, and I was successful to develop methods that could very easily detect mutant rice, even up to 3 copy number. So after being successful in rice and tomato, we are expanding the trial to other crops like potato. So now, just to give you an overview of what Darwin project is, I will just show a video about the project, and maybe you will get to know more about how we are using knowledge for mutant lines and wild type to provide consumer a fair food system.
As human beings, we have evolved to adapt to new world conditions and survive. So too has nature. Every organism is then bound to transform its DNA through spontaneous and advantageous changes called mutations. This is the natural origin of genetic evolution, and this is what new genomic techniques try to replicate by inducing designed mutations. However, the current scientific and technological barriers in detecting NGT products that comply with European legislation are causing ambiguity for consumers, food producers, and agri-food operators. Here is where Darwin Project inserts. Darwin is an EU-funded project that aims to create more sustainable and fair food systems through 3 main goals. 1, validate detection methods for NGT genetic changes in plant products. 2, 2. Use AI and bioinformatics to obtain complete genetic fingerprints of NGT products. 3. Create a data space, a blockchain to increase traceability and transparency of NGT products. How? 2 different plant species, rice and tomato. 3 realistic scenarios will be played out to validate detection methods for seeds and pure products, mixed seeds, and processed food. This will help advance research, increase trust in technology, improve knowledge in society, and support economic growth, all the while ensuring traceability across the food chain and transparency in legislative framework. Do you want to follow food systems evolution? Keep an eye on our social media and website.
So, by that, I would like to end my presentation quoting the fact that in the battle against plant disease, knowledge is our greatest weapon. Thank you for your attention.